Welcome to Animal Biotechnology. Today, we are diving into the microscopic world of genetic engineering to explore the delivery vehicles that make it all possible. Imagine trying to deliver a fragile package to a secure factory. You need a reliable truck. In biotechnology, our package is a gene, the factory is a living cell, and our trucks are called vectors. We will explore three essential types of vectors: plasmids, cosmids, and phagemids. By the end of this lesson, you will understand how each vehicle works, their cargo limits, and how to calculate their efficiency. Let us get started. Now that we understand the need for delivery vehicles, let us look at the most fundamental one, the plasmid. Think of plasmids as the basic delivery trucks of the laboratory. They are small, circular, double-stranded DNA molecules that exist naturally in bacteria. Why do we use them? Because naked DNA cannot just be dropped onto a cell. It would be destroyed or ignored. Plasmids safely carry our gene of interest through the cell membrane. Once inside, they ensure the gene gets copied. They are incredibly easy to work with, stable, and can be purified in massive quantities, making them the absolute foundation of modern molecular cloning. Building on the idea of plasmids as delivery trucks, let us examine their specific parts. A plasmid has three critical features. First is the origin of replication, which allows the plasmid to copy itself independently of the main bacterial chromosome. Second is the multiple cloning site. This is a specialized region where we use restriction enzymes to easily insert our target gene, much like loading cargo into the back of the truck. Finally, there is a selectable marker, usually an antibiotic resistance gene. This acts as a tracking device, allowing us to easily identify and isolate only the cells that successfully took up the plasmid by growing them in a toxic antibiotic environment. Having discussed the components of a plasmid, let us visualize them on this standard vector map. You are looking at a circular schematic representing the double-stranded DNA of a plasmid. Notice the three distinct colored regions. The blue arrow represents the origin of replication, labeled ORI, which drives the copying process. At the top, the green segment is the multiple cloning site, or MCS, where our foreign gene is inserted. At the bottom, the red arrow shows the antibiotic resistance gene, labeled AMP-R, which serves as our selectable marker. This simple circular design is what makes plasmids so effective for routine cloning of small DNA fragments. Now that we know what a plasmid looks like, we need a way to measure how successfully our plasmid trucks entered the host cells. This brings us to the transformation efficiency formula. Conceptually, this formula scales up our experimental results to tell us how many successful deliveries we would get per microgram of DNA. T represents the transformation efficiency, N is the number of successful colonies counted on the plate, M is the mass of plasmid DNA used, V sub T is the total volume of the cell suspension, and V sub P is the volume actually spread on the plate. If the number of colonies increases, our efficiency increases. If we use more DNA but get the same colonies, the efficiency decreases. Let us apply this formula to a concrete laboratory example. We are given 0.05 micrograms of plasmid DNA, a total suspension volume of 1,000 microliters, and a plated volume of 100 microliters. We count exactly 250 colonies. We need to find the transformation efficiency. We chose this formula because it accounts for both the DNA mass and the dilution factor of our plating. First, we substitute our known values into the equation. We divide 250 by 0.05 to get 5,000 transformants for the plated portion. Then we divide 1,000 by 100 to find our dilution factor of 10. Multiplying these gives our final answer. This means we achieved 50,000 transformants per microgram, indicating a highly successful delivery process. While plasmids are excellent for small jobs, our previous calculations assume the cargo is light. What happens when the gene is simply too large? If you try to stuff massive DNA into a standard plasmid, it becomes unstable and gets deleted by the host cell. To solve this, scientists engineered Cosmids. If a plasmid is a small delivery truck, a Cosmid is a massive, heavy-duty cargo ship. They are hybrid vehicles designed specifically to carry massive loads of genetic material safely. Cosmids are incredibly important for constructing genomic libraries, where we need to represent the entire genome of a complex organism. Without them, mapping genomes like the human genome would be exponentially more difficult and expensive. To understand how these heavy-duty cargo ships carry so much weight, we must look at their hybrid nature. A Cosmid combines the best features of standard plasmids and the lambda bacteriophage, a virus that infects bacteria. The magical ingredient is a cohesive end site, known as a cos site, derived from the virus. This molecular signal allows the entire Cosmid, along with a massive piece of foreign DNA, to be packaged into an empty viral protein shell. Once the viral shell injects the DNA into the host cell, the Cosmid circularizes and replicates exactly like a normal plasmid. This clever trick allows us to bypass the physical limits of standard plasmid delivery. Because Cosmids rely on viral packaging, we must understand the physical limits of the viral head. This leads us to the capacity formula. Conceptually, the viral head can only hold a specific, rigid length of DNA before it overflows. S sub insert represents the maximum size of the foreign DNA fragment we can add. S sub phage is the absolute maximum capacity of the lambda phage head, which is physically limited to about 52 kilobases. S sub vector is the size of the empty Cosmid vector itself. If the size of our empty vector increases, the available space for our target gene strictly decreases. If the total combined size exceeds the limit, the packaging process will fail completely. Before we attempt to clone a massive gene, let us see this capacity limit in action with an example. We are given a commercial Cosmid vector named PJB8, which is 5.4 kilobases long. The lambda phage head has a strict upper limit of 52.0 kilobases. We are asked to find the absolute maximum size of the animal DNA fragment we can insert. We use the capacity formula to subtract the vector size from the total viral headspace. Substituting our values, we subtract 5.4 from 52.0. This simple subtraction reveals our strict upper boundary. The final answer is 46.6 kilobases. Physically, this means if you try to insert a gene complex larger than this, the DNA will not fit into the viral head, and your experiment will yield 0 colonies. We've seen trucks and cargo ships, but what if we need a vehicle that can change its behavior on demand? Enter the Phagimid, the amphibious vehicle of cloning. Imagine a car that transforms into a boat at the push of a button. Most of the time, a Phagimid acts just like a normal, reliable plasmid that is easy to copy and store as double-stranded DNA. But when we give the host cell a specific biological signal, the Phagimid switches modes. It starts behaving like a virus, producing a completely different type of genetic material, pure, single-stranded DNA. This dual-purpose nature makes Phagemids highly versatile for advanced genetic engineering tasks. To understand how this amphibious transformation happens, we must look at the Phagemid's unique architecture. A Phagemid contains two completely different origins of replication. It has a standard double-stranded origin from a plasmid and a single-stranded origin from a filamentous bacteriophage. Under normal conditions, bacteria use the plasmid origin. However, if we superinfect the bacteria with a helper phage, which provides necessary packaging proteins, the Phagemid switches to the phage origin. It begins producing single-stranded copies of the inserted DNA, which are secreted out of the cell. This is absolutely crucial for techniques like DNA sequencing and site-directed mutagenesis, giving us the stability of a plasmid with the power to generate single-stranded DNA on demand. Once we have triggered our Phagemids to produce single-stranded DNA, we need to know exactly how much we have recovered. We calculate this using a spectrophotometer, which measures light absorbance. Conceptually, the more DNA present, the more light is absorbed. In this formula, C represents the final concentration of the single-stranded DNA. A sub 260 is the absorbance reading at a wavelength of 260 nanometers. The number 33 is the specific extinction coefficient constant, calibrated only for single-stranded DNA. D represents the dilution factor used when preparing the sample. If the absorbance reading increases, it directly indicates that the concentration of DNA in our original tube is proportionally higher. Let us put this concentration formula to work with a final laboratory example. We are given a sample where 2 microliters of purified DNA are mixed with 98 microliters of water. The machine gives an absorbance reading of 0.45. We need to find the actual concentration of the original tube. We use this specific formula because it incorporates the constant for single-stranded DNA and accounts for our sample preparation. First, we calculate the dilution factor by dividing the total volume of 100 by the sample volume of 2, giving us 50. Next, we multiply the absorbance of 0.45 by the constant 33, and then by our dilution factor of 50. The final answer is 742.5 micrograms per milliliter. Physically, this high concentration means we have successfully generated enough pure single-stranded material to proceed with complex downstream sequencing. We have covered a tremendous amount of ground today, so let us reinforce the most important ideas. To master genetic engineering, you must choose the right vehicle for your cargo. Plasmids are your everyday delivery trucks, perfect for routine cloning of small fragments up to 10 kilobases. Cosmids are your heavy-duty cargo ships, utilizing viral packaging to transport massive genomic fragments up to 45 kilobases. Finally, Phagemids are your versatile amphibious vehicles, offering plasmid stability with the unique ability to generate single-stranded DNA on demand. Understanding these distinct advantages and physical limits is your first critical step in designing successful biotechnology experiments. You now have the foundational knowledge to navigate the world of cloning vectors.